Bismillahirrahmanirrahim. Uh, Assalamu alaikum, dear student. Welcome to the course of Pharmacology and Therapeutic. I'm Fawad Ali, Assistant Professor, Department of Pharmacy. Today we will discuss about an antibiotic group that is macrolide. Here are some outlines. We will discuss one by one, like the chemistry of the macrolide, um, the and then the prototype drug like erythromycin, its antibiotic spectrum, mechanism of action, resistance, pharmacokinetics, therapeutic uses and adverse effects, and its drug interactions. So the chemistry of the macrolides, they have a macrocyclic lactone ring, this is known as a macrolide ring, to which a deoxy sugars are attached. The macrolides, the drug included in the macrolides are the erythromycin, clarithromycin, azithromycin, roxithromycin, spermycin. General structure, they all have three characteristic parts in their molecule, like a macromolecule lactone, a ketone group, and a minor deoxy sugar. In some of the macrolide, a neutral deoxy sugar attached to the aglycone ring, like the micro, which is also known as a macrocyclic lactone ring. So this is basically a macrolide basics nucleus. If you see, the the structure is in the structure, like there are two main groups, substituents, like the R1 and R2. If we changes the like in the erythromycin R1, there will be a methyl group, and R2 there will be a simple hydrogen. But in a clarithromycin, the R1 and R2 both have a methyl group in it. So this is a basic structure. Basically, the macrolide, the first, macrolide include the erythromycins. Basically, the erythromycin are isolated from the streptomycin erytheus in 19, which is a natural source. Other than erythromycin, like erythromycin, azithromycin, all are basically the semi-synthetic. So general properties of the erythromycin, which is a prototype drug, is a pure base, poorly water soluble, but soluble in organic solvent. It has stability at 4 degrees centigrade, lose activity in acidic pH or at 20 degrees centigrade. Activity is basically enhanced in the alkaline pH. Dispense as an esters or salts, we'll discuss later. So the spectrum of the antibacterial spectrum of the erythromycin is basically a bacteriostatic, but may be bactericidal in high concentration against the very sensitive organisms. So basically, it is a bacteriostatic. Effective against aerobic gram positive cocci and bacilli like Streptococcus pyogenes, Streptococcus viridans, pneumococci, Staphylococcus, Clostridium perfringens, Corynebacteria, diphtheria, Listeria monocytes. They are basically effective in against most effective against these gram positive cocci and bacilli. They have less active gram negative organism, including activity against the gram negative organism like the H influenza and Neisseria meningitis. They also have an activity against the Neisseria gonorrhea, Rhodiola protusis, Campylobacter jejuni, and Helicobacter pylori. Before going to start the mode of action, we'll have an animated we will see some animated videos related to the mechanism of action. For example, for example, DNA shown here is a double-stranded DNA bacteria. Like DNA of all the living organisms, they contain the unique genetic codes of all the protein required for the bacterial survival, reproduction, growth, and metabolism. They also code for three different kinds of the RNA required for the protein synthesis, that are the ribosomal ribosomal RNA, which is also known as rRNA, messenger RNA, mRNA, and transfer RNA is also known as the tRNA. In the process of the protein synthesis, double-stranded double-stranded DNA is unwound and separated in a region which code for the specific protein that is required. So one strand of the DNA serve as a template for this process known as the transcription. Transcription result in the formation of the messenger RNA. Once the strand of the messenger is complete, it will detach from the DNA template and in turn attach with the ribosomes, which have bacterial ribosome have two subunits, 50S and 30S. 50S is the largest subunit and 30S is a smaller subunit. Synthesis A synthesis of the polypeptide chain begin at this step. This include this step involve the aligning of the transfer RNA molecule. Each transfer RNA molecule carry unique amino acid determined by the sequence of the transfer RNA, which when aligned along the messenger RNA ribosome join together to form the polypeptide chain. This step is known as a trans, translation. Ribosome continually add amino acid to the polypeptide chain until it reaches the point along the messenger RNA. Signal it to stop at this point. Ribosome releases the protein molecule. So proteining the releases which is the finished protein molecule. So this is the basic idea of what protein how the protein synthesis involve different steps. So it is important because macrolide mode of action, macrolide act as an inhibitor of protein synthesis by binding with this 50S ribosomal subunit, resulting the inactivation or decreases the ability of ribosome to synthesize the polypeptide chain. Why? Because the polypeptide chain, the synthesis of polypeptide chain begin with the transfer RNA, or because of the translation. So macrolide basically inhibit the translation step of the protein synthesis. So now we will here read this mode of action like bacteriostatic, but bactericide for some sensitive marker enter bacteria by the passive diffusion, bind with the receptor at the 50S ribosomal subunit, inhibit the translocation step, so inhibit the elongation of polypeptide chain. May also interfere with the formation of the initiation complex activity, which is shown in the next slide. Activity enhanced in the alkaline. This is the initiation, like the messenger RNA formyl methionine plus transfer RNA. This is an initiation complex. So it basically interfere with the initiation complex. No peptide bond found. So this is the like the mode of action. Like this is the normal initiation complex plus the different transfer RNA, addition of the different amino acids and the polypeptide chain result in the polypeptide chain. But here in macrolide, it interfere with the initiation complex, resulting in the inhibition of the the the, the complex, this initiation complex. So like this is another slide which elaborates the mechanism of action of the erythromycin. Erythromycin and clindamycin bind to the 50S subunit, thus inhibited in the translocation step. This is inhibiting the translocation step. There is no translocation. Ultimately, the polypeptide chain is also inhibited. So other microorganism binding to the 50S ribosomal subunit of the sensitive bacteria and inhibiting the proteins. There are some bacteriostatic other bacteriostatic drugs that most bind with the 50S subunit: macrolide, chloramphenicol, lincomycin, clindamycin, quinapristine, and the linezolid. So there are some drugs that most bind with the 50S subunit. So resistant, natural resistant erythromycin normally naturally some to the staphylococcal, pneumococcal, and streptococcal. They develop resistance. Some are the like acquired, reduce the permeability of the cell membrane because of the efflux high efflux of the drug by an active pump mechanism. Methylase enzyme lead to the ribosomal modification and inhibit the binding of the drug. Hydrolysis by the esterase is normally produced by the enterobacteriaceae. So ribosomal protection, alteration of the 50S ribosomal protein by the chromosomal mutation like Bacillus subtilis, Campylobacter, gram-positive cocci. And cross-resistant, like for example, cross-resistant means if the if the organism is required is an acquired resistance toward the erythromycin, so it would be resistant to the erythromycin and azithromycin. So complete in whole group, they are mostly cross resistance. Kinetics of the erythromycin is an adequately absorbed from the GIT tract. It is destroyed by the gastric acid and hence must be administered normally as an entire coated tablet to protect it from the gastric fluid. Food may delay the absorption. It widely distributed in the body and reaches the therapeutic concentration in the prostatic secretions, but does not cross the blood-brain barrier. It is partly metabolized in the liver, excreted in the bile, and undergo enterohepatic circulation. So normally there are different type of erythromycin, like erythromycin bases, erythromycin esters, like estolate or N-ethyl succinate, propionate. They are the more different esters of the erythromycin. Like estolate as a lipid-soluble acid stable product undergoes hydrolysis in blood and tissue after absorption. Ethyl succinate mix double ester products of the erythromycin, better oral absorption as compared to estolate, and oral suspension for the children without the bitter taste, and then propionate lipid soluble esters of the propionic acid. They have more better oral absorptions. So then there are some erythromycin salts like erythromycin stearate, erythromycin lactobionate, erythromycin glucosipate. There are different drugs, depend like for example stearate, lipid soluble salts of stearic acid, better oral absorption. Acid sensitive entire coated form is an acid stable in small gut base exchange occur. Free erythromycin release. Erythromycin lactobionate water soluble. They are the water soluble salt of the lactobionic acid for the parental dosage form, and it's a glucosipate siptate water soluble salt of the glucoheptonic acid for the parental dosage form. Distribution. So they are distributed readily into the intracellular fluids. Activity can be achieved at all sites except the brain and CSF. Poor penetration in the synovial fluids. Protein binding is 70% concentrated in the liver. Elimination half-life of the erythromycin is 1.5 hour. They cross the placental barrier, highly concentrated in the prostatic fluids, liver, and bile. They are metabolized by specifically by the CYP3A4 microsomal cytochrome P450 enzyme, eliminated as an elimination. They are mainly eliminated in the bile as an active metabolite. In bile, concentration may be 50 times that of the plasma. Undergo enterohepatic circulation lost in feces. It's an inactive metabolite excreted in urine only 2 to 5%. Not removed by the dialysis. Do not need to be adjusted in the renal patient. So because they are not removed by the dialysis, better oral absorption and longer T half than most of the roxithromycin and clarithromycin, etc. They have more oral absorption as compared to the roxy, clarithromycin, and azithromycin. So this is a summary of the pharmacokinetic. Like do not penetrate into the CNS, do not cross the blood-brain barrier. Metabolite of erythromycin and azithromycin appear in the bile, while the clarithromycin appear in the urine. This is mostly a difference in the excretion of the different macrolides. So therapeutic choices of the erythromycin include the drug of choice for the coronary bacterial infection like diphtheria, sepsis, mycoplasmal infection like pneumonia, yeah, like Legionella infection causing pneumonia, chlamydial infection specifically in the respiratory, eye, genital, and newborn, pertussis, like mostly in the whooping cough, community acquired pneumonia as a penicillin substitute in the penicillin sensitive patient like tetanus, staphylococcal infection, syphilis, and streptococcal infections. Is also effective in the prophylaxis of the endocarditis prior to the dental. Prophylactically, we use in normally when we are doing the dental procedure. So prophylactically, we use the erythromycin to reduce the risk of endocarditis. Prophylaxis of the rheumatoid fever in the penicillin allergic patients, Helicobacter pylori. Mostly we prefer like erythromycin, mostly in the erythromycin, most commonly in the H. pylori irrigation therapy, mostly in the triple and quarter regime. Most commonly add the erythromycin. Doses of the erythromycin is orally 250 to 100, 500 milligram QID 4 times a day. Children 40 milligram per kg per day, and IV for zero point 0.5 gram 8 to 12 hour uh, hourly per day. Adverse effect GIT. Most common adverse effect includes GIT upset like nausea, vomiting, anorexia, oral administration, diarrhea. Basically, erythromycin, specifically erythromycin, the drug specifically erythromycin macrolide is responsible for the diarrhea, which is because of its mode of action. It is also known as the gastroparesis. Yeah, it is also known as the gastroparesis or prokinetic agents, which increases the GAT motility. They have the specific receptor in the GIT, which is known as the mortalin receptor. Erythromycin, by binding with the mortalin receptor, increase the GIT motility, resulting in the diarrhea. Liver toxicity. Istolate salt have the more than four, if you use 14 days, it cause hepatitis, fever, jaundice, liver impairment, abnormal LFT liver functions, appropriately as in hypersensitivity reactions. So if we uses the acetylate salt of the erythromycin for more than 24 for more than 14 days, resulting in the hepatitis. Other hypersensitivity reaction includes the fever, eosinophilia, and skin eruptions. Cardiotoxicity. So erythromycin causes the cardiotoxicity with the QT prolongation, especially in the patient underlying cardiac diseases. Some studies suggest, some study reported confirmed a link between the erythromycin and ventricular tachyarrhythmias, your ventricular arrhythmias, causing a sudden death in patient. If patient that prolong the metabolism of the erythromycin like vancomycin and diltiazem by interfering with the CYP3A4 enzyme. So if this that's why the combination of the different drugs like the calcium channel blocker like verapamil and diltiazem with the erythromycin, resulting with the QT prolongation and even within the certain death. So there are some erythromycin have the microsomal enzyme inhibition. For example, it have inhibited the CYP1A2 enzyme, increase the oral anticoagulant concentration in the body, like warfarin. It also increases the concentration of warfarin, like oral anticoagulant in the body, resulting in the if any the concentration of the oral anticoagulant like warfarin increases, resulting in the bleeding. Theophylline lower therapeutic index by inhibiting the CYP1A2, methylprednisolone CYP3A4, utilizing it by for its metabolism. So the amount of the methylprednisolone increases, and that's the case with the cyclosporine. They utilize the CYP3A4 enzyme for its own metabolism, increases the amount of the cyclosporine and methylprednisolone, also increases The oral increases the serum level of the oral digoxin. It is also a lower therapeutic index drug, resulting in the ventricular arrhythmias due to its increased viability. It's also so the concentration of the or antihistamines like terfenadine and estimazole increase the risk of arrhythmia. The same is the case with the calcium channel blocker. Like there is a link between the erythromycin and ventricular arrhythmias in patient in the patient that prolong the metabolism of the erythromycin. This is what the terfenadine, estimazole, and verapamil and diltiazem interfering with the CYP3A4 enzyme, and this prolonging the metabolism of the erythromycin, resulting in the QT prolongation. Thank you. Any question regarding this will be most welcome. If you can ask through the email, your question will be welcome.